Methane is a greenhouse gas 25 times more potent than carbon dioxide and an important driver of climate change. One of the main sources of methane emissions? Cow burps. Communities of microbes called microbiomes exist in the guts of humans, cattle, and other animals. Microbiomes have many important roles, from helping digest food to preventing infection and regulating the immune system. But in cattle, they also do something else. They make methane gas, which the animals emit as burps. And yes, they've studied it. The methane is mostly coming from the front of the cow.
The reason that we really want to tackle this is because from the pre-industrial period, we had 1.1 degree centigrade increase, and we want to try to keep it up to 1.5. Out of the 1.1 degree centigrade, 0.5, almost half of it, is because of methane. If you look at the anthropogenic sources of methane, about 35% is from livestock.
While many other sectors like energy and transportation have technological options for reducing emissions, agriculture has proven to be a tough nut to crack. Ermias, Kebreab, and Matthias Hess are IGI researchers based at the University of California, Davis. They're working on a novel approach to reducing methane emissions, editing the microbiomes of cattle. Their inspiration comes from an unusual place: red seaweed. Previous work from the pair has shown that feeding cows red seaweed can dramatically reduce their methane emissions. The seaweed works by blocking some enzymes that microbes need to make methane.
Basically, they were collecting a lot of different types of seaweed from Australia, and they were just looking to see which ones reduce methane emissions. We did the first study in dairy cattle in vivo, so in real cattle, and we saw a substantial reduction, over 60% reduction in methane emissions.
Some studies show methane reductions as high as 98%. Now, the pair is working on using CRISPR genome editing to target the same methane-making genes within the genomes of microbes that live in the cattle's gut. The goal is to create a safe, inexpensive, one-time treatment that would permanently reduce methane emissions and could be used by cattle farmers around the globe.
The ideal outcome of that project really would be that we could alter the microbiome in a cattle really early in life as a calf, and then that microbiome would remain stable and would produce very little, if no, methane at all. So we're not touching the animal, we basically change only what these microbes can do.
Imagine if you could give every young calf a simple treatment like a pill, and it would reduce their methane emissions for life. To get there, the IGI teams at UC Davis and UC Berkeley are working to understand the genomes of methane-making microbes.
In my lab, we look at the microbiomes, get the DNA out of these organisms so we can look at different genetic material that is present in them. The next step is that we give that information to our collaborators in Berkeley. They will develop some of these tools that they will give back to us here in Davis.
The next step would be moving to field trials, where they can test if the results found in the lab can be reproduced in the gut microbiome of the cattle.
What we would like to see is, do we get a stable microbiome? How much methane emissions is there? Have you reduced the emissions significantly? What are the microbes that are in play? But ultimately, the main thing is, do we have methane or not?
For a problem as tough as climate change, we need to use every tool in the box to target greenhouse gas emissions at every level, even from the tiniest sources hiding inside cows.